All right, everybody, if you can find your seats, if you're here in the room, that would be great. Thank you so much, everyone, for being here today for our seminar. We're really excited for today's speaker, who's a familiar face to many of you in the room and possibly some of you online. Just a reminder, this is a hybrid event, so we have folks online and folks in the room. My name is Cinnamon Moffett. I'm the Research Program Manager here at Oregon State University's Hatfield Marine Science Center, and I will be your host. Roseanne is our volunteer. Thanks, Roseanne. She's going to be the individual monitoring questions as they come into the room via chat. And for those folks that are online, we do have your cameras, mics, and screen shares disabled at this time, but we do hope you ask questions. Just put those into the chat, and Roseanne will read those out when we open up for question and answer. For folks in the room, couple of reminders, again, because this is a hybrid event, if you have a question when we get to the question and answer, just raise your hand and I will bring you this mic so that folks online can hear your question as well. Or you can go to the mic stand, which is right over there if you happen to be up there at the upper bleachers, or you can watch me run. You just have to wait for me. But please ask for a mic when you ask the questions and then everyone can hear. For folks in the room, I also ask that you sign in for two reasons. That helps us justify buying cookies. It also lets our speakers know how many folks were in the room for their outreach numbers. So just a reminder there. Okay, I've got a couple of really quick announcements and then we'll get this thing started. I wanted to let everybody know or remind them of Marine Science Day. So our flyers just came off the presses about 2 hours ago. So very excited. If you don't know about this event, this is our open house that we do every year. About 2,000 folks come in and explore all of the research that we have here. For my researchers in the room, if you haven't told me you want an exhibit, please let me know soon so we can hold a table for you. For everybody else, hope you can join us on the 13th. It's a pretty exciting day. I also wanted to talk quickly about next week's seminar. So we're very excited to have Dawn Barlow, who is a postdoctoral scholar here with the Marine Mammal Institute, talking about the work that she's been doing the last year or so. So she's going to be talking about what 3 decades of nearshore surveys reveal about habitat, distribution and abundance of gray whales and harbor porpoises in the Northern California current. So we're really excited to see what Dawn has to share with us. So I hope you can join us next week on March 14th. I also just found out this morning that we had a last-minute cancellation. So I have an opening for a seminar on April 11th. If anybody has somebody they want to volunteer, please let me know. I'd be happy to have somebody help us out with a seminar. Seminars, we love to have our speakers on site, but if somebody can join us remotely and that's the only way they can come, let me know and we can work with that too. So April 11th, I've got an opening. Okay. But the reason you're all here today is for today's speaker. Bryan Black is currently an associate professor at the University of Arizona Laboratory of Tree Ring Research. But before that, as I was saying, he was here at Hatfield from 2003 to 2012. He has a bachelor's in biology from Westminster College. He has his master's and his PhD in forest resources from Pennsylvania State University. His work applies dendrochronology techniques in growth increments formed in the hard parts of marine and freshwater species, including fish, bivalves, and coral. He uses these to establish long-term patterns in productivity and their relationships to climate and long-term and the impacts of human activities. So Bryan was originally trained as a forest ecologist, and he maintains interest in developing histories of forest dynamics, disturbances, natural disasters like landslides and earthquakes here in the Pacific Northwest. And so we're very excited to have him back with us to share. I also, so I don't forget, wanted to remind everybody that Bryan will have office hours tomorrow. So if you are interested in continuing this conversation, he is around and will be able to speak with you. But without further ado, I'm going to hand this off to Bryan.
Thanks very much for that introduction, Cinnamon. And I'm really delighted and honored to be here at the Hatfield Marine Science Center. It's been several years since I visited. I think I haven't been on campus since 2015, so a lot of change and exciting to see the new facilities and visit some of my familiar haunts as well. And as Cinnamon mentioned, I'm based at the University of Arizona at the Laboratory of Tree Ring Research in Tucson, and continue to apply and work on techniques that I developed while here at the Hatfield. Okay. Let's see. There we go. That gets it started. And, when I started, I've been reflecting a bit on when I started here 20 years ago now, back in 2003, the summer of 2003. I had just finished my PhD in forestry and specialized in the ecology and development of Appalachian forests, and had no idea that organisms, such a diversity of organisms, could live a long time and form rings just like trees do. And when I started here, I was interested in fish, but also clams that form increments. The world's oldest animal is a clam at 507 years, Antarctica Islandica, that was pulled out of the North Atlantic. But when I began, was interested in rockfish, like the split-nosed rockfish, which learned at the time how to section otoliths on, say, the low-speed isomet saw, which I bet is still here, this isomet, and get the thin sections. And whenever you thin section these otoliths, the cross section looks a whole lot like a tree. And could we apply then tree ring techniques to the growth increments in these long-lived marine organisms? And critically, if you're a tree ring scientist, you spend a lot of time doing this process known as crossdating to ensure that all increments are exactly placed in time and accounted for. And so this is based on the idea that as climate varies over time, it induces synchronous growth patterns among all individuals of a given species and locations. So in the desert Southwest, precipitation is usually very limiting to tree growth. So you get a drought, all the trees will form a narrow year. You get a moderate rainfall year, they form a relatively wide year. Another drought, a narrow year, maybe a very favorable rainfall year, they all form a wide ring. So as climate varies over time, it sets up this synchronous growth barcode, synchronous growth pattern among these trees. And cross-dating is the process of matching those patterns to ensure that all the growth increments have been identified. So in this schematic, you might have 3 trees that were cored in 2009, and we know that the ring under that bark was formed in 2009, and then we can match patterns back through time toward the center of the trees. And say if you happen to overlook an increment in one of those tree cores, then the growth pattern would be offset in that core a year relative to the growth patterns in the other trees. So you'd immediately see this frame shift and know that you'd made a mistake, and could go back and identify where that mistake occurred and correct it. And in that way, you're sure that you've assigned the correct calendar year of formation to every tree. And by the time you do this to 40 or 50 trees, you really learn that pattern and also have very high confidence that you've accounted for all the rings and have everything correctly aligned in time. And so the big thing was, can we cross-date in fish? And turns out, yes, we can. We get this kind of flight recorder of environmental conditions that the fish is recording in its otolith from the year of its first year of life on out to capture. And you can even see from where you're seated this year-to-year variability in growth, like this heavy white band, which is a very narrow increment formed during an El Nino year, and another narrow band formed during another El Nino year. And so finer scales of variation or patterns of variation I can use in those otoliths to cross-date them and then measure them and develop and extract growth information where each line is measurements from a different otolith. And you can see they all have this age-related growth decline that can be easily removed. We're not real interested in this age-related growth decline, but getting rid of that biological effect so that we isolate environmental variability. And that can be done just by fitting, say, negative exponential functions to model that age-related growth decline, and then simply divide the observed measurements by those predicted by the age-related growth decline to remove that age-related decline and start to really enhance the shared synchrony that these fish have that's going to reflect the environmental conditions they experienced. And so you can detrend all these fish, and then here's the residuals that show lots of ups and downs over time, can be averaged together into then a master chronology for population-wide variation in fish growth over the last few decades here for the central part of the California current. And then once we have these records, can investigate, well, what climate variables are reflected in this growth chronology, and thus are influencing growth of these fish. So we have this really long time series of growth, and what are the climate drivers that might tell us about productivity for these fish that are providing kind of a top-down look at the ecosystem. And of course, here upwelling being a big driver to explore, where the winds from the north are moving surface waters offshore in combination with the rotation of the Earth, lifting deep, nutrient-rich cold water to the surface that supercharges phytoplankton and drives productivity. And so we can get records of upwelling, say here month by month, because it may be that certain months of the year months affect an entire year's worth of growth for fish. And we see this in trees quite frequently, where high elevation especially, you get a warm spring, the trees get a jump start on the growing season and have a wide ring. And then during a cold spring season, have a delayed start to a cold spring delays the start of the growing season, and you get a narrow ring. So it's something analogous happening in the ocean. So we can say, take February upwelling, compare it to our chronology, and get a correlation, and then repeat this for upwelling records for every month of the year, and even some from the year prior, and begin to see these peaks in correlation, a seasonality profile, which isn't quite what I expected, where we get these peaks, say, in the winter, especially February. Right now, January, February is a time of year that we see a lot of sensitivity to upwelling. And again, could be you get this very robust upwelling in the winter months at a time of year it might not otherwise occur, and it could give the fish a jump start on the growing season to form a wide ring. But winter is critical. And what's great about these chronologies is we know they're exactly placed in time, so you can relate them to other biological records. So it's possible to develop other fish chronologies or relate them to other biological indices, such as seabird reproductive success as recorded in the Farallon Islands since 1972 off San Francisco. And so here we have egg lay date. Earlier lay date indicates more favorable ecosystem conditions, and also fledgling survivorship. The more survivors you have, it's a better indicator of, more survivors means more favorable ecosystem conditions. So I have time series in the birds and the fish, and it turns out that they all covary as well, and can be summarized if you say take their common interval in this period outlined in the red box, extract the leading principal component, what's the dominant pattern that they all share. It explains 59% of the variance in the whole data set, and unsurprisingly correlates very strongly to winter upwelling. So that we have the seabird reproductive success and the rockfish independently corroborating one another on the importance of this wintertime upwelling, wintertime ocean conditions in Central California. That's enabled by developing these long chronologies from otolith increments. So as and that relationship between winter upwelling anomaly and that leading biological principal component being quite compelling. Now, as a tree ring person, then I'm interested in bringing this back onto what's happening on land. And it turns out that atmospheric pressure during the winter months is very closely tied to upwelling. You get, say, these are these high pressure systems. Then their clockwise rotation helps drive those northerly upwelling winds. And this is the correlation between winter upwelling and gridded sea level pressure over the northern Pacific, northeastern Pacific. And not only do these high pressure systems drive upwelling, they also block the onshore transport of precipitation. It deflects the storms to the north and then can induce drought on land. And so this brings in trees then into the equation. And so that winter North Pacific high, if we just extract North Pacific high off of California, or the North Pacific sea level pressure off of California, it correlates very strongly to upwelling at all these locations where upwelling is measured. Also sea level, winter sea level up and down the coast. River discharge on land because of that link to precipitation. And also significant correlations with precipitation. Winter precipitation is measured in all these red regions on the map. And because a lot of trees are very sensitive to precipitation, then we have this North Pacific high index that relates also to trees, and especially blue oak, which are exquisitely sensitive to wintertime precipitation. And if I graph out all those time series, flipping a few just to illustrate synchrony, you can see that rather remarkable covariance across marine and terrestrial systems, all dancing to that tune of the North Pacific high off of California. And this is something that's very biologically relevant to the rockfish and the seabirds, and is also captured by these blue oak chronologies. And the blue oak go much farther back in time at recording this very key wintertime climate indicator for the region. And so we can use then blue oak to give us a much longer history of California, of this biological indicator for the California current. And in fact, we can superimpose the average of those blue oak chronology on top of the sea level pressure indicator, and also sea level is measured at San Francisco. You can see great covariance, and a scatter plot of those two, where the blue oak growth anomaly is an excellent predictor of the North Pacific high that's causing these variations in upwelling that's so important to rockfish and seabirds, and then gives us now this much longer history of that key climate component of the California current, which is quite well dominated by high frequency variability, especially ENSO-related variability. But what's intriguing, I think, is that some of these negative values that would be associated with El Nino conditions have been increasing in frequency and intensity in the recent decades. And these arrows indicate some of the most extreme negative anomalies that would be kind of associated with El Nino conditions, very poor rockfish and seabird growth, but the trees would do great with the precipitation. But that this variability is increasing. There's a trend of rising variance in this time series, and also a trend of increasing extreme events, especially in the recent decades. And that we see this in rising hydroclimate volatility here on the West Coast in the winter months. And we've been hearing a lot about climate whiplash, and that you can see that in these tree ring records that are that reflect in a very important biological variable for the rockfish and the seabirds. And that the Earth system models, the CMIP6 models, would indicate that this climate whiplash should only increase in the future for the winter months for the California region and up into Oregon. And of course, this has been published in recent years. This work with the rockfish that I began here at the Hatfield Marine Science Center and continue to work on with various projects now, especially integrating across chronologies. And beyond the fish, I thought I'd also mention a bit about clams, because they too form growth increments and live a long time. The Pacific gooey duck being one of my favorites, that's really gets quite long-lived and abundant from Puget Sound on up through Kodiak, Alaska, and support a multi-million-dollar fishery. So there's a lot of collections of gooey duck around. And what's we can use gooey duck for is climate histories of the northeastern Pacific. And that's an area where there's a lot of uncertainty, especially in the tree ring record. Here's several different tree ring reconstructions of North Pacific sea surface temperatures. The black line is the Pacific decadal oscillation, which is an instrumental indicator of past sea surface temperatures of the North Pacific. You can see this really bad or this poor agreement among these tree ring records, which is a bit of a well-kept, now not so well-kept secret in the tree ring community, that there's not a lot of confidence as to what Pacific variability has been like prior to 1900, because we just don't have good agreement among the tree ring records. And well, can the gooey duck come in and provide some more clarity on this past of North Pacific temperatures? And I've developed a number of chronologies now using modern shell that's live collected. And some of these sites pick up a really great regional climate signal, especially this northern site, the Tree Knob Islands. And the thing is, the gooey duck live 150 years, which is a pretty long time if you're a clam, but it's really not that long of a time frame if you're interested in past climate. And so what we have to do then is get divers out. And so there's a whole Underwater Harvesters Association. It's been very gracious about going out and excavating pits into the ocean floor and sucking out these gooey duck from the mud. And the deeper you go into these pits, the farther back in time, and collecting dead shell for me. Here's my first shipment of it that they send down to my lab in Arizona, where we then process these shells. We cut out the midsection, embed it in epoxy, get it on a high-speed saw, cut it so right through the middle, and then polish it very well. We do what's called an acetate peel in this hinge region. to acid etch the rings, which helps us see them better. And this is what the ring structure looks like in the geoduck. And then can this be a history of North Pacific climate that could, tell us about temperature variability? And so what we've done then is gotten hundreds of these shells and just cross-dated them, worked out this big puzzle. And these are now the intervals spanned by geoduck, where each black bar is the lifespan of one geoduck that we've been able to piece together through cross-dating. And this was round one of geoduck, and now I have a second from the National Science Foundation, where we're trying to do 1,000 years continuous, but have this great history now of from geoduck growth increments that relates well to temperature. And so we can show this as a chronology. So we have a continuous, absolutely dated chronology from the 1720s to present, and then some snippets of well-replicated chronology in the past. And the blue line is an instrumental record of sea surface temperature for the British Columbia coast. And you get an idea of how good of a relationship there is between the proxy and the instrumental record. And this geoduck chronology also relates quite well to tropical corals, low frequency variability in tropical corals. One's a sea surface temperature reconstruction within the red box, and then the green diamond and the blue diamond are other coral records. And the geoduck track the low frequency variability in those corals, which gives me confidence for picking up a really robust Pacific-wide pattern of low frequency variability. And so the point now is, can we reconstruct North Pacific climate from geoduck? And the target being this leading principal component of gridded sea surface temperatures for the northeastern Pacific. This is what the loadings look like in the map. So you get this very strong coherence of correlation along the coast of the Northeast Pacific, and the so-called named the arc pattern because of this arc of strong correlations. And the time series of this arc pattern is shown as the red line in the time series figure. And shown also, here's the PDO, but the Pacific Decadal Oscillation, the linear trend is removed, so you don't get this warming trend, but that is retained in the arc pattern. So the target is this dominant pattern of sea surface temperatures in the northeastern Pacific, which is the red line on the graph. And we can add that on to our geoduck chronology and start to see the geoduck tracking that northeastern Pacific arc pattern, in addition to the more regional temperatures in blue. So that's the geoduck, but trees are also picking this up. And is there a way that we can use the geoduck now, that really long record, to help us screen the tree ring chronologies? And so here's our geoduck, and then all the available tree ring chronologies for Western North America and Eastern Asia are in gray dots. And it turns out the geoduck correlates very strongly to a nice swath of those. Everywhere there's a red dot is where there's a strongly significant and stable relationship between geoduck and trees. And what's interesting is if we plot the correlations between geoduck and all the tree ring chronologies, we get this scatter plot where on the x-axis, I guess I'll say that the patterns become very interesting whenever we look at the stability of that correlation between geoduck and trees over time. So we divide the data in half and say that, so we have the early half of the data set from the 1726 to 1858, and then from 1859 through 2008 being the other half. We can show here on the x-axis the correlation between geoduck and trees in that early half of the data, pre-1859, and then the difference in correlation between the recent and the early half of the data. And what we see is shifts in the tree ring and geoduck chronology relationships over time, where trees that used to be negatively correlated with the geoduck have now shifted in the positive direction, and that trees that used to be positively correlated with geoduck have shifted in a negative direction. So there's this change in the climate growth relationships of trees that's probably related to long-term warming and these shifts between temperature and precipitation limitations in trees that having this long geoduck record reveals. But there's also a core group of trees, these red dots, which are common in the map and on the biplot, where we have strong, stable relationships between trees and geoduck, and that those are the trees we should be looking for that would have stable relationships to North Pacific temperatures. And so we can identify those tree ring chronologies. And then here's what they look like. They go back even farther than the geoduck. So there's our geoduck screened tree ring chronologies for their stability and strength of relationship with temperature, and average those together. And we get a nice reconstruction with an R squared of 62% of the variance between the temperature record, ARC SST, and trees that goes back over the past few centuries. And what we can do too is screen tree rings or tree ring data on a variety of criteria. You can say, for example, those that relate really well to ARC SST, those relate well to the early half of geoduck, those relate well to the latter half of the geoduck, and do an ensemble approach to the reconstruction. And so then where each black line is a different realization of that ensemble and the uncertainties associated with it that give us still a rather consistent idea of what the past variability has been like, with the geoduck in blue superimposed on top and illustrating that coherence across the marine and terrestrial systems. And shows us overall then this variability over the decades from warm to cool regimes, and now culminating in this ultra warm regime of heat waves here above, say, 2 standard deviations above pre-industrial, and the frequency of those, and especially the warmth that we've seen since the 2010s in the blobs. This provides a really nice a history of the lead up to these heat waves we've been experiencing and how unprecedented they are, as has been in the news so much, and that I'm sure this crowd is very acutely aware here on the West Coast. And over the years has been great to develop, with myself and colleagues around the world, now a global network of chronologies of fish and bivalves. And it's exciting now to do comparisons across them and syntheses. One of the big goals for the geoduck now, after we work on this 1,000-year chronology, is what's the linkages to the North Atlantic or the tropical Pacific, and how is it that the high frequency variability like ENSO originating from the tropics gets mixed in the water column of the North Pacific and buffered to emerge as this lower frequency interdecadal variability. And then if we have 300, 500-year records from the tropics, we can test those hypotheses to see if we just say add autocorrelation to the tropics, does that then show up with the patterns we see in the North Pacific to test this kind of capacitance that the North Pacific water column might have, and these linkages across broad regions of the Pacific. So that's exciting now that we're getting these networks and can do broader scale comparisons and go farther back in time. Another thing I'd talk about today too is that trees not only tell us about climate, but also natural disasters in the Northwest. So that some of these trees that are useful for the North Pacific temperatures are also useful for dating, say, major earthquakes. And with projects that the seeds were planted for, say with Bob Ziak here back in the early 2000s of looking for evidence of Cascadia activity or earthquakes. And this has been recently a big topic, say, in the Puget Lowlands area, where Washington, is being, where the Oregon Coast Range is migrating north and compressing Washington against the immobile mountains of British Columbia, and that this compression has caused a series of faults, of shallow crustal faults around what is now Seattle, Tacoma, and Olympia. And there's a series of them. There's a Seattle fault zone right through Seattle. There's the Saddle Mountain fault zone in the foothills of the Olympics, the Tacoma fault zone, and also an Olympia fault zone. And radiocarbon dating from various organic sources shows that these faults were all active about 1,100 years ago, but the uncertainties span, in most cases, more than a century. So did these faults all happen in very close time interval, or was it one fault 1 decade and then several decades later did another fault go off? Were earthquakes separated by 100 hours or 100 years? That's a really big difference in understanding the hazard. And so the question is, can trees help narrow those uncertainties, especially in the Seattle fault and the Saddle Mountain fault to start with? And it turns out there's a lot of wood material. Here's some that's even had slid off a hillside into Lake Sammamish, and there's another 2 or 3 landslides in Lake Washington where entire hillsides with trees slid in during these earthquake events. And divers have collected wood from those trees that slid into the lakes. There's also in the Saddle Mountain Fault, thrust up 25 feet to impound a stream that formed a lake that flooded a forest, and that lake and those trees are still there. And so we organized an outing with divers from the U.S. Geological Survey and a big underwater hydraulic saw that we had divers go out and collect wood from that lake, which was quite the adventure. While we sat on the dive platform, they were out, it'd take 45 minutes to get a wedge with absolutely no visibility. It was quite an impressive accomplishment, but got that wood. And so get it back to the lab, and I was able to cross-date it all together across those sites. And so we could see that across 3 different sites where we had the most wood, that the growth, the trees all cross-dated among one another, indicating they all lived and died at the same time. And they also cross-dated across sites, indicating that those trees across sites also died the same time. So that across both faults, those trees died the same year, and also with a completely formed ring, such that they died sometime in the dormant season, the winter, across both faults, indicating that both faults ruptured at about the same time, either in one massive fault that was linked or in very rapid succession over the course of maybe hours to months, as happened in Turkey about a year ago, those terrible earthquakes. One about and here it would be about a 7.4 and a 7.5, or combined as a 7.8. But then so what's the exact year? And what we could use then is a new technique in high resolution radiocarbon, where about 10 years ago, a scientist in Japan was dissecting individual tree rings and running them for their radiocarbon, and discovered this really big pulse in radiocarbon from the year 774 to 775. And it's from what must have been an enormous solar storm of the likes of which we've never seen anything in modern times. This in fact would really fry our satellites. You'd see aurora in the tropics. I mean, it would be a massive solar storm that would cause this level of carbon-14 in the system. But it provides a very distinct time-specific pulse. And if we can, and we're lucky that these trees likely lived through that event. And so we could narrow down and find out about where we thought this event occurred, and dissect out the individual tree rings. And here's my colleague Charlotte Pearson working at helping dissect out some rings and measure the radiocarbon. And lo and behold, we found the spike. And there's the big pulse in radiocarbon right at 774. And when we knew where that pulse in radiocarbon was, then we knew exactly the absolute dating of these trees, and could find out that the end date was in fact the winter of 923 to 924. So exactly 1100 years ago, right now, there was a massive single earthquake or rapid 1-2 punch in what's now Seattle, Olympia, and Tacoma that killed all these trees across a wide number of sites in the region. And they all have a complete last ring that indicates, it happened sometime in the winter. Now the question is, and by the way, we also found a long chronology up on Vancouver Island, about 1100 years or 1300 years in length, that hadn't been published before, that we could slide back and forth against our earthquake chronologies and see that the peak correlation, this t-value is just a correlation, but of all the possible lags in time, that at 923, we get this really big spike in correlation, indicating an independent verification that 923 was the date, and that we had this massive event that killed trees across all these sites. So the tree rings could really dial in now the accuracy of, or narrow down the uncertainties associated with these earthquake events. And that just came out in September and got a bit of press. I think next now is to tie down the Olympia fault zone. And so we have wood from the Olympia fault zone near this little Skookum site. Only now we deal with much uglier wood, these stumps that we have that we can sample for radiocarbon and put it on a radiocarbon curve here and narrow down the date to 927 to 948 that we're excited about, and see if that also, if we can lock that down. But the problem there is that the ring widths aren't that useful. Ring widths, you know, and some of these sites, they don't covary real well. But it turns out that if you measure the auction 18, sometimes that's much more covariable. Here's an example of some pines from near Bend, Oregon, where the ring widths, they have a lot of, they don't tightly covary as the auction 18. And so here's the ring widths from a couple of our Puget Sound sites. The ring widths, but if you do auction 18, the correlations get much stronger. And so we're hoping that this will help us lock in, say, this uglier wood from the Olympia fault zone into Tacoma. And I also have hopes that this might help us too in the Cascadia Subduction Zone, which needs no introduction, where the dating for the Cascadia Subduction Zone all comes from the northern part of the Cascadia near Willapa Bay, these ghost forests of trees that were killed in the earthquake and tsunami. And in the 90s were dated to 1700. But it turns out that, you know, just the northern part of Cascadia could have ruptured and sent a tsunami to Japan, as was, you know, helped to nail down the date for this earthquake. The whole thing didn't need to rupture, so we don't know for sure whether all of Cascadia actually ruptured in 1700 or not. But it turns out there's other tree ring sites down here at the Mad River and Eel River in Northern California. And so we went out and sampled this Eel River last summer and have sites, you know, these stumps, and collected a bunch of roots. And then here's what the geology looks like, you know, where we think there's a tsunami, and then some overlayer of channel sand and silt, and get these sampled and back to the lab. And then hopefully auction 18 data might help us cross-date these too and compare them against a 1,000-year isotope record from redwoods, and might provide new information on if Southern Cascadia ruptured in 1700 also. And on top of that, we just found the Mad River Slough samples at a warehouse in Seattle, and they're being shipped to my lab as we speak. The last thing I'll mention too is you know, new dating techniques here with Oregon relevance on Mount Mazama that formed Crater Lake. This is one of the largest eruptions of the last 10,000 years. Spread ash in this pink area clear out to Yellowstone and up to Alberta. Massive event that shows up in the Greenland ice cores. And anytime you're working with stratigraphy in the Northwest, onshore or off, this is a major marker. But it happened about 7,600 years ago. And the question is, can we narrow it down? And I started looking online for places where you might find wood that was buried in Mazama, and found this Lomolo Lake might be a site that some amateur naturalists had found. And last summer went out and rented a boat and looked around, and you can start to see this wood sticking out of the ash deposits. And went out and scoped a bunch of sites last summer and found a bunch of wood that might be associated with Mazama. And later brought a crew from my lab out, and also someone from the Cascades Volcano Observatory. And we found all kinds of wood, especially after the water had lowered. And it's all this really carbonized wood. It was buried in pyroclastic flows and superheated and turned to charcoal almost instantly. And that's where it's remained. So we filled up an entire Ford expedition and took it back to Tucson, and have you know, now sanded this and got the rings resolved, what the charcoal looks like. And check out the bark on this one. You can see the bark where the red arrow is. That's the final ring. So this tree formed bark and was killed instantly by Mount Mazama, and still has that record of its very last ring. And in fact, there's 4 trees with the last ring. Here are all the measurements I've cross-dated out of it. And now the question is, what's that exact calendar year those trees died? And what's that final ring? And at my tree ring lab, we have this history, this 8,000-year record of bristlecone pine. And so I was able to dig out this piece of bristlecone pine that lived about the time we think Mazama erupted. And what we're going to do is sample ring by ring this bristlecone pine that was absolutely dated, and measure its radiocarbon, and then pattern match that radiocarbon and the bristlecone pine against the radiocarbon pattern in the Mazama wood to lock down that exact date. So we dissect here, and measure the radiocarbon. We're getting a radiocarbon lab at the Laboratory of Tree Ring Research, this building. We're converting part of the second floor. It's being shipped now to do this high resolution radiocarbon. I think our first application will be to date Mount Mazama, and also work quite a bit, I think, with a lot of future work to be done in Cascadia with wood sites up and down the coast here. Also archives of wood from the last quake in 1700, and a potential to get much more exact dates of earlier earthquakes in Cascadia as long as we can find the wood. So a lot of neat stuff that would come up, and I'm planning to do a sabbatical this fall and spend a little more time out here on the West Coast to look for some of this wood and just continue that work of, you know, ecosystem linkages where the trees represent the terrestrial to the rivers, to the near shore and offshore, and the integrations across them that we can do with growth increment techniques and cross-dating and getting everything aligned in time. And, you know, there's many people I've worked with on this project, but I think, you know, for this acknowledgement, I'd like to really acknowledge my thanks, you know, to the Hatfield Marine Science Center, especially George and Susan Bollert and all my colleagues here, you know, who've been so instrumental in their support and mentorship and friendship over the years in launching my career, not only professionally, but also personally, which I'm very grateful for. And so glad to be back to provide this, to share these updates, and looking forward to continuing to be a member of this community in the future. So with that, I'll wrap up and take any questions.
There's some familiar faces on those pictures.
Yes.
All right, everybody. I think there was lots in that presentation. So let's open it up to questions. Let's start with any questions online, Roseanne. Okay, for folks online, go ahead and put it into the chat and we'll work through those questions in the room.
Using the chainsaw underwater sounds pretty difficult. Do you ever use some sort of mechanical core to sample trees, rings?
Yes, the increment borer is good, you know, a hollow shaft of metal with a bit on the end. That's good for live trees. When they die and get rotten, it's much more difficult to use the borer. So saws are the way to go for dead stuff.
All right, other questions in the room? Okay, thank everyone.
Yeah, Bryan, good talk. I'm wondering, have you ever had an opportunity to look at any archaeological sites like Indian middens in the Northwest region for possible otoliths or shells that you might be able to date?
No, I haven't. You know, the gooey duck are usually too deep to show up in middens. So I haven't found at least for gooey duck that middens are of use. But yeah, it may be a possibility for other shell species. So far, it's been most effective just to go out and have divers collect them to get the best material. But it's certainly worthwhile to keep an eye open for what might show up in middens.
I'm going to hand the mic down.
Hey, Bryan,
Hey, Bob.
great talk. Welcome back.
Thanks.
Hey, great question about the middens too. I've always wondered about those because they're right on the bluffs, and you know the idea the tsunami came through in 1700. I've always wondered how old those middens were, and if that were related. But that's the side question. Actually, my main question is: it's neat you're seeing those correlations in Eel River for the 1700 earthquake. Is that limited to the coastline, or you actually see some evidence of the big earthquake back like in the Olympics, where you're working for around Puget Sound? Because that's, you know, a common model is that this big earthquake would have a lot of impacts in the Willamette Valley. So I wonder if they're actually showing up in the tree rings.
Yeah, right. So, you know, if trees aren't killed, as you know, it's hard to see these signatures of the earthquake. But the other thing I'd be interested in would be just inundation from the tsunamis. We've talked a little bit about this, but it's possible that carbon-13 might make might shift rather dramatically after the tsunami, and that's what we see in pines from Japan. Some Japanese researchers have documented this big spike in carbon-13, and we see that preliminarily just right here at Mike Miller, that right around 1700 there's a big spike in carbon-13, and that's something that would be useful to explore as an indicator of inundation. But beyond that, you know, trees don't seem to bat much of an eyelash during these earthquakes unless they're really killed or somehow knocked over during the event. But they just shake it off. It's really quite amazing, as we've seen.
Right.
It's
That's a good point about,
yeah, they just really seem able to sway and not be too bothered.
One more question since I have the
Yeah.
microphone. Really cool that you had that 1,000-year hindcast, basically, with the geoducks.
Yes.
So have you looked at machine learning models to actually do forecasts, for temperature and see if there's some kind of pattern you can pull out of that?
Right. So the future, you can query the CMIP6 models, the Earth system models, the coupled model intercomparison project. So that's something we'll do is query that area of the Pacific, and then under various scenarios, what do we see through 2100? Under different emission scenarios, I guess societal pathways they call it now.
Right. Okay. Cool.
You know what you get.
Thanks.
All right, hang on.
Bryan, thanks very much for that talk. I just want to offer up, when you're out for sabbatical, we have very few confirmed observations of geoducks. They do occur here, down on the Oregon coast. But do you think it'd be valuable to add gaper clams? We'd love to have you work on our gaper clams and figure that out and see if there's any value in chronology in that group, because they're very abundant here, and they're also buried and preserved as death assemblages.
Right. If they live a few decades, then that could be very useful.
Yeah.
It's fairly long lived then.
Yes.
Okay. Yeah. I'd like to talk more about
let's
that.
talk about that. Then
Yeah,
the
I
other one
think
I
I've
wanted to
seen
ask you
those.
about, we've recently aged some of our sea urchins and have sea urchins up to 200 years old here. I don't know if they would have value because they disintegrate.
They have something like a tooth or
Yes,
something in
yeah,
the that's
So let's talk about that too. Yeah.
okay.
All right, other questions in the room? Other questions online? We're gonna let Bryan off this easy. Okay, there we go.
Afterwards, we got one more.
First of all, great talk, amazing work. Have you ever thought about, or would it potentially be possible to do something with short-lived species? For example, there are excellent records of scales for sardine and anchovy in the Santa Barbara Basin. And they are, they were sort of, it's thought to be environmentally driven with booms and busts from 1700, basically. I think that those scales would be available. And I'm just wondering if you might be able to tie in, those short-lived species with environmental variables. Like maybe they even have, differences in their growth rings over that short period of time, and over a longer, maybe a 30-year record, you might be able to do something along those lines. Again, I know it's off the wall, but I figured I'd ask.
No, excellent point. I have two thoughts on that. One is, yes, the short-lived are useful, especially if they're collected over time. So I worked with a group from the University of Iceland and the Institute for Marine Research in Norway with their cod collections. And so there's cod collections that go back to 1900. And they had a big project funded to do, say, 100-year chronologies from cod based on this continuous collection of, the species that lives maybe 7 or 9 years. But you have hundreds and hundreds of cod collected every year. You can piece together these chronologies for multiple places in the North Atlantic. So if you have these long collections, then that's totally possible. And second of all, for the Santa Barbara Basin, I was on a PhD committee where a student had taken those cores and dug out those otoliths. He'd even taken a tree ring course I taught, or a sclerochronology course, and was interested in the rings, but he only had time to count and ID all the otoliths and his cores from the Santa Barbara Basin. But the next step was going to be, can we section those and look at changes in growth or chemical or isotopic composition to tell us about really long-term changes over the last several thousand years. And that step has not yet been taken, but I think would be quite interesting. Yep, that's a great idea.
So I have a novice question. Can you talk a little bit more about why on the tree rings, some of them do not correlate? Like, why do we need to look for other patterns?
Yep.
So I can understand why in a fish, but I'm having trouble in a tree.
When trees are really happy and are growing in favorable conditions, they're not very much limited by climate. They're just forming ring after ring after ring about the same width over time. If trees are growing in stressful conditions, then they respond quite dramatically to climate and they have a very strong barcode. Here in the Northwest, trees are pretty happy. And especially if you get into a situation where there are roots and that growth is distorted because of the buttressing from roots and distorted growth that accompanies roots, then it becomes extremely difficult to cross date from width. And so that's when we want to start calling out the next wave of technologies by measuring the oxygen 18 or carbon or radiocarbon in the roots and see if we can cross date that. And if that's possible, then that brings a whole new generation of techniques to wood samples that no one's been able to work with in the past, but now could really revolutionize what we understand about these earthquakes and past events, because it's those uglier pieces of wood that preserve the best and have the rings. So if we can get past ring widths and get to the oxygen 18 and the high resolution radiocarbon and get that level of precision that we're used to in tree rings, that would be a big deal. And I think I'm so excited to try that out, because in the past that covariability has been more just a novelty, but now we have a real application for it of trying to pin down past earthquakes.
Nice. Very cool. All right, last call. Any questions online? Any questions in the room? Bryan, when are your office hours tomorrow?
I think from about 9:30 till 3.
And you are in the library Barry Fisher room.
Barry Fisher room if anyone wants to drop by.
Perfect. All right, let's give our speaker a round of applause, please.
Right. Thanks, everyone.
All right, everybody. I hope to see you next week. Remember Marine Science Day, and let me know if you have a speaker for April 11th. Thanks, everyone. See you next week. Okay, for folks online, we're going to end the presentation now. Thanks so much for being here and hope to see you again in the future.